Hey again, guys. Part 4 in this 5-part series on photosynthesis and the biochemistry of photosynthesis. We started with a broad overview, and then in the last video we really started narrowing it down and looking at the light reactions, and specifically something called non-cyclic or linear electron flow. What we're going to do now is we're going to see why photosynthesis requires also some cyclic electron flow, and I'll show you exactly what that cyclic electron flow looks like. Okay, so non-cyclic and cyclic electron flow. Let me get this little picture out of the way here because there's no good place for it. So you know that says non-cyclic and cyclic electron flow. Okay, so in this image we can see a thylakoid, right? One of these little stacked guys. It's all green to represent all of the photosystem 2 and photosystem 1 components that have all of their antenna arrays in them. We know that light hits P680, or it actually hits the antenna complex, which shuttles energy to the P680 reaction center on the surface, the stroma surface of photosystem 2, the complex of proteins in photosystem 2, launching a pair of electrons from P680 up into a very high energy state, which are then grabbed by a series of carriers that shuttle them through all the way till ultimately they're handed off to photosystem 1 at P700. P700 being the reaction center in photosystem 1. In the process of these electrons moving through, and there's 2 electrons being moved, as they move through the cytochrome complex, this cytochrome complex actually has a pump associated with it, a proton pump. And so for every 2 electrons, every pair that moves through, 2 protons are going to be moved from the stroma into the lumen. So out here in that feels like a cytoplasm kind of space within the chloroplast, into the actual lumen within the thylakoids. At the same time, we know that the electrons that were taken from P680 have to be regenerated, and so P680 takes them from H2O, which should be plentiful, should be plenty of water around. When it hydrolyzes that water, when it splits it apart, maybe hydrolysis isn't the best term, but when it lyses, it's photolysis actually, that water, it results in O2 and protons being produced. So for every H2O, you get half of an O2 molecule. So for every 2 times this happens, you get a full O2. And for every 1 time this happens, you get 2 free protons that weren't there before, also in the thylakoid lumen. So for every pair of electrons moving from P680 to P700, we concentrate 2 protons in the thylakoid lumen, and at the same time remove 2 protons from the stroma of the chloroplast, and we're creating a gradient across this membrane. And we know that those proton electrochemical gradients can be used to power ATP synthetases. Now these electrons make it to P700. The protons then can run through an ATP synthetase, they mean the same thing, and power phosphorylation from ADP to ATP, just like we saw in cellular respiration. But we're not done, because we said that really what we need to make is both ATP and NADPH. And so now the light hits our P700 complex, or our photosystem 1 complex. The electrons, or the energy from the photon of light, bounces around within the antenna complex until it reaches the P700 reaction center, which kicks the electrons up into a high energy state. And there they get carried by ferredoxin to NADP plus reductase, which then just simply hands them to NADP plus, keeping the electrons in a relatively high energy state in the form of NADPH. So now, here in the stroma of the chloroplast, we've created 1 ATP and 1 NADPH for every pair of electrons that moves through. So let's think about that for a minute. This means that ATP and NADPH are produced in a 1-to-1 ratio. However, they're consumed by the Calvin cycle, the dark reactions, which we haven't talked about yet, out here in the stroma. They're consumed in a 3-to-2 ratio. So for every 3 ATPs that are used in the dark reactions, only 2 NADPHs are used. So what does that mean? It means you're going to run out of ATP before you run out of NADPH, right? So imagine you've produced 300 of each, right? Because they're produced in a 1-to-1 ratio. You've got 300 ATPs and 300 NADPHs. By the time you've used all 300 ATPs, you still have 100 NADPHs left. And that's just going to continue. And so your NADPH pool is going to grow and grow and grow as you go through this whole process, and it's going to be way out of whack. So plants, and particularly chloroplasts, have adapted a really cool mechanism called cyclic electron flow that they can use when their ATP pool starts getting really low relative to their NADPH pool. So they can bring the ATP back up again, right? Because ATP is getting consumed faster than the NADPH, and so we want to bring it back up again so we don't end up with this huge surplus of unusable NADPH. So ATP and NADPH are produced in a 1-to-1 ratio, but they're consumed in a 3-to-2 ratio, and that's why cyclic electron flow is necessary. I'll explain the mechanism of cyclic electron flow in a second. First, what you need to do is hit pause and make sure this makes sense to you. Don't move on if the why doesn't make sense. Okay, enough said. So what does cyclic electron flow look like? Cyclic electron flow, here's our photosystem 1. We know that P680 kicked electrons that it stole from water up to a primary acceptor. It runs through this electron transport chain on its way down to P700 in photosystem 1. And as it passes through the cytochrome complex, it activates a proton pump that moves protons, and then that gradient that it's generating of H+ ions across the thylakoid membrane is used in a chemiosmotic way, just like in cell respiration, to drive ATP synthesis, which we call photophosphorylation as opposed to oxidative phosphorylation in cell respiration. Then P700 receives another photon of light, jumps the electrons up into a high energy state. The primary acceptor grabs them. Now here's where the decision is made molecularly. If there's still plenty of ATP around relative to NADPH, these electrons will continue in a linear non-cyclic fashion to flow until they rest in NADPH, meaning they started in water, they got kicked to a high energy state, ran through one electron transport chain, got kicked a second time into a high energy state, ran through another electron transport chain, ended in NADPH. And we got one ATP, one NADPH. If, however, the electrons here get kicked from P700 to the primary acceptor, if ATP levels are getting low relative to NADPH levels, ferredoxin, this shuttle here, doesn't have to give those electrons to NADP plus reductase. It can, in fact, cycle, that's where cyclic electron flow comes from, these electrons back to the cytochrome complex. And remember, every time they pass through the cytochrome complex, we pump 2 electrons across the membrane. And so we pump 2 more electrons, and they get kicked up again. We pump 2 more electrons, and they get kicked up again. We pump 2 more electrons, and they get kicked up again. And the more electrons, I keep saying electrons, we're pumping protons. The more protons we pump, the stronger the gradient across that membrane, and we can keep making ATP. Until the ATP pool catches up to the NADPH pool, in which case ferredoxin will be signaled through internal signals to stop giving the electrons cyclically to the cytochrome complex and instead get back to non-cyclic electron flow and give them to the NADP plus reductase to get back to making NADPH. So that's what cyclic electron flow is all about. So we talked about the why, because they're being used disproportionately to the way they're being made. Therefore, we run out of ATP sooner than we run out of NADPH. It becomes cyclic flow because the same pair of electrons can continuously get kicked up into a high energy state at P700 in the photosystem 1 and then cycled back to the cytochrome complex rather than in a linear fashion being given to NADP plus reductase and NADPH. And every time they go through cytochrome complex, they pump more protons, we increase the gradient, we get back to making more ATP. So hopefully that makes sense to you. Remember, we're talking about photophosphorylation, and we're talking about chemiosmosis. The chemiosmotic theory applies here just like it did in cellular respiration, where you accumulate protons on the inside, the lumen of the thylakoids. You deplete the protons outside in the stroma through the cytochrome pumping, and then that gradient is used to drive an ATP synthase that makes ATP by now what we call phosphorylation. So cyclic and non-cyclic are working together to make sure that there's enough ATP and NADPH to support the Calvin cycle, which is our next video. So I hope that this has been helpful. If it's confusing, if it's difficult, watch it again, pause at key places, rewind where you need to, make sure you understand this content before you then move on to the Calvin cycle.